All right, I'm going to do a quick poll. Who in the room has a system suitability method for their instruments? Kinda. We've got like some kindas, right? No one's like, yeah, I totally got it. No. Okay. What I'm going to show you are tips and tricks to setting one up and the whys of having a system suitability. So when I was in graduate school, and I went to grad school specifically to learn how to do mass spectrometry, and I worked on a very old FTICR system that took a lot of time and effort to get the ions from where they were being generated at the ion source to the pending trap or the heart of the FTICR, and there were not focusing quads or anything, there were just lenses that you had to tune with voltages, and you would either see a signal or you wouldn't in the beginning. So instead of tuning for a signal, you were fishing for a signal, and then once you had it, you had to tune. And so going through this process would take a couple of hours, and then you would run your sample. And I had a sample that had gone through a couple of days preparation of just labeling and cleaning up. And when I went to analyze the sample, I didn't have any signal, and I was like, man, clearly that mass spec must be broken, right? You don't get a signal, it's the mass spec's fault. And I didn't have an HPLC in the thing either, so that's what I thought it was, and didn't think it was my sample or anything. And as it turned out, that kind of was the case. I took it to another instrument in the building, and whoa, amazing signal, because that one was tuned up and done properly. Now, the mass spec itself is a system, but what many people are using here is more integrated with the chromatography and the mass spectrometry. So normally we can tune and calibrate the mass spec, you can tune and calibrate the flow meters or the flow rates or the pressures or do leak tests on the LC, but when you put them together, there's that little weak point of what may or may not be happening there. So system suitability is designed to test the entire system, the LC and the MS and their integration point. So what is system suitability? It's an integral part of testing all of the bits and pieces of the system, the hardware, even the software to some extent. And it allows you to check the system performance at any point in a very longitudinal way with different users. It helps you track if there are any changes that are caused by just normal wear and tear, or maybe your vendor decided, hey, we've got a new version of instrument control software, and when you install that, everything goes to crap. It happens, but you don't really know it happens if you're always changing the analytes you're monitoring, or if you have lots of different users in the lab using the instrument. So, simply put, like after all these technical definitions, analysis of a known sample to assess system performance. And this is actually real data we acquired and plotted in Skyline in the very early days, where we had multiple labs running the same kind of analyses. They were all given the same sample. And this is the profile, mass spec intensity versus retention time for a bunch of transitions for one peptide. And this is what you would expect to see, a symmetrical Gaussian chromatographic peak, good intensity. The same sample performed on a different system, clearly there's something wrong with that, right? Not only is it visually asymmetric and tailing, but it's a lot less intense than the other peak. Unfortunately, it's not always this obvious. So here's a question. And the answer's there. Don't know why I keep doing that. Is system suitability the same as QC or quality control? And the answer is no. So system suitability is not, if you run many different chromatographic gradients on your system, if you use your mass spec to do data-dependent acquisition and data-independent acquisition and PRM, having a method set up for those parameters and those different types of methods would not be considered system suitability. System suitability is a method that does not change over time so that you can compare your system performance when the instrument was first installed right before it needs to be cleaned, right after it's been vented and cleaned, before and after software changes, before and after changing a column, any kind of point in time. But the point is, you can't compare what the system performance looks like if you are tweaking things along the way, right? So a QC is something that is very method specific. It could also incorporate sample handling. If you do digestion, you might have a QC for the digestion procedure. And this QC will change from analytical purpose to analytical purpose. So you might have a QC for data-dependent runs. You might have a different QC for PRM and targeted runs. Whereas system suitability, the point is you want to try to make sure you are grabbing or utilizing all aspects of the analytical instrument the way you normally would, and that can be applied generally to all the methods that you normally use. Whoops. Okay. When should you run a system suitability evaluation? Pretty much anytime you don't want to lose data, you want to run it before and after. But you're not going to bracket every single sample with a before and after because that decreases your throughput. So what's good to do is to set up a system suitability method if you don't have one, and then get a baseline of your instrument's performance. You look at things like peak area, peak symmetry, signal to noise, how the data look, peak separation from the chromatographic standpoint, and so on. And then from that point on, once you have a decent benchmark, you just run it anytime you can, instead of letting the system sit idle over the weekend. If your queue stops on like a Sunday afternoon, queue up some system suitability runs. Keep the system going. Particularly with NanoFlow, these LCs like to stay pumping. And then also, you want to make sure you do it when you think your system isn't performing well. You know, you sometimes get that feeling like, oh, that result that I got for my data-dependent run, it wasn't exactly what I thought it was going to be. Let me look at the last system suitability run that ran before it, and I'll run one right after it and see how they compare. I bring up the software because these instruments are, delicate analytical instruments that are like hunks of metal with voltages applied, that have really tight specifications on measurements of, the electrodes and the hunks of metal inside, and everything is controlled by software. So you press a button on the computer and a series of things happens over on the instrument. And he's not here, but anyone who's a Skyline daily user knows, if you ask for a feature to be included in Skyline and then there's a daily release, every now and then there's another daily release an hour after that first one in Skyline, because sometimes when you implement a change in the software, you break something over here. So keep in mind that software upgrades you should take them with caution, and always make sure that you're doing testing before and after, because it controls your instrument. Okay, what are the metrics of a system suitability protocol? What are the things you want to monitor and keep track of? So if you're looking at an LC-MS system, we break it down into the two components. For the MS, we look at the signal response, which relates to sensitivity, the background noise, chemical noise. Oftentimes, if you have problems with, like on a triple quad, there are electrometers that can cause high background signal. And so your peak may normally be, 100, but if the background is now at 50, your peak is half the height. And that's an electronics problem and a hardware problem. Looking at the SRM transition ratio, this is something that, again, we know that peptide fragments very reproducibly in ion traps, in collision cells. And so if you see changes to that pattern and their intensities, then something could be happening. I personally have accidentally replaced my empty argon cylinder that I used for collision gas with helium because I was tired. I'm always tired though, so it doesn't really count. They're the same color, and you think that I would have seen this label that's on this like 5.5 foot cylinder, because I had to roll it down the hallway, so it must have passed by my eyes like 50 times before I installed it, and it was helium. And helium is much smaller than argon, and I wasn't getting any MS2 spectra. I was loading my sample, I was seeing like these little blips in my gradient, like, what the heck is going on? So I went back to my system suitability and I ran that, and it was not performing, and I tracked it back to what was the last thing I did after a good system suitability, and it was, oh, I ran out of argon, so I changed the tank. And so I ran an MS1 scan, which does not require the collision gas, and all of my peaks were there. And so I was like, oh, shit. I went over and I looked at the tank, and like, it's helium, and this is after I started making a fuss, and I contacted the engineer, and they did their tests online remotely, and they're like, something's wrong with your collision gas. And I'm like, yep, nope, it's fine, I got it taken care of, we're good now. And I admitted to my mess up. So for the chromatography, reproducibility of retention time is very important. And not just reproducibility, but the absolute amount. If all of your peaks are coming out 20 minutes later than that same method run in your friend's lab, then something may be wrong and the system is not running optimally. Even if they're reproducible, they also have to make sense in the context of the method. Peak shape, they should be symmetrical, although there are some peaks that might have modifications that cause them to act a little bit different, but those would probably not be great for system suitability. The full width half max of the peak is important to pay attention to. Chromatographic resolution, so this is how closely or separately your peaks are spaced, and this can change column degradation or if you're using the wrong column type or different than you're used to. And then carryover, there can be things that accumulate in your system over time that just require cleaning or just upkeep to minimize anything that would jeopardize your system performance. So this is the chromatographic resolution. These are two peaks that are very similar in shape. And in this scenario, they have a chromatographic resolution of 6.2. Resolution in chromatographic terms, the bigger the number, the more separate they are. And there's an impact not just on retention time, but also on the peak width. So to calculate chromatographic resolution, you use retention time difference divided by half of the width of one peak minus the width of the other, so the difference. So when we go from here, which might have been acquired in May, and here, this might have been acquired in June, you see that the peaks are broader and they're coming closer together. So if you see this with your system suitability sample, it will probably impact the data that you're trying to do for real science. Not that this isn't real science, but it's just less exciting. So as part of the CPTAC initiative back in 2008, I think, this paper came out, and it was looking at performance of the systems across a series of labs for discovery LC-MS proteomics. This was published by Paul Redditz in MCP. This should be freely available. And they designed software that looked at all these different metrics for MS1 chromatography, at the ion source, dynamic sampling for doing these data-dependent MS2s, the quality, and lots of metrics from the MS2s. Now, this is very overwhelming, and it may not need to be this involved. And so since this paper came out, there's been a lot more focus on paying attention to different things in your data that can help tell you if things are running well and can help to diagnose when things are going wrong, specifically what those things are. So for those of you that don't have a system suitability protocol in place, one question is, well, what can you use? If cost was no option and you wanted to make sure everything was great all the time with regards to your sample, you would want to purchase something that is QC'd by the company you purchase it from, and they can guarantee its integrity. And they've done stability testing, and they can assure you that it's fine with freeze thaws. It lasts at auto sampler temperature conditions for a week or a month, and they can give you that information. If cost is an option, you can set up evaluation of something for your system suitability. It just takes a little longer. So if you're going to do that, you need to make sure that it's stable with time. So what I do is I set up a sample and I put it in the auto sampler and I just inject it a couple times a week once a day, depending on what the sample load's looking like, and I track its performance, assuming the system is running fine, and that the only thing that would be changing during that time is the sample itself. Of course, you may not be lucky, and you may have some other issues with the system that's causing a change in the signal or the chromatographic peak shape. So starting and baselining system suitability with a new sample is challenging, which is why if you can start with something that's already gone through this process and it's been validated by a company, that's the safer way. Then what you can do is carry overlapping experiments where you can transition to some other system suitability as long as you perform the analyses on the two different samples at the same time. That allows you to transition from one sample to another. Because the diagnosis retention time standards are a commonly used system suitability sample. Bovine serum albumin digest, cytochrome C digest. These are all things that are reasonably cheaply purchased. For data-dependent things, the HeLa lysate digest or any lysate digest that people can grow themselves and purify and aliquot and have in large quantities are all completely valid. It's just you need to make sure that they are stable. So if you do make them yourselves, it's kind of on you to make sure that they are good. The idea would be if you have your systems are dedicated to proteomics analyses or peptides and protein analyses, and you have separate systems for small molecule, then I'd recommend you have different system suitabilities for these things. If you have systems that have to serve multi-functions, like I have my instruments right now have all of our HPLCs on carts, and we'll have nanoflow set up one day, and then 2 days later we'll have to switch over and run high flow. And so I can't use the same system suitability if my LCs are changing. So I may have a few different system suitability methods depending on what my LC is or what the mass spectrometer is. You won't have the same system suitability method on a triple quad as you will on a high-res accurate mass instrument. So ideally you'd like it to be similar to your actual samples. If you're doing intact proteins, then it makes no sense to look at resurfing, right? If you're looking at small molecules, then I wouldn't suggest looking at BSM. You want something that has minimal carryover and is not exactly like your sample. If you're studying human cell lines, then maybe you want to use something that's from yeast. Inexpensive because you're going to be using it a lot, or you should be using it a lot. Easy to prepare, so you don't want something that you have to go through a whole day process to make enough samples for a few weeks, because that's just too time consuming. Ideally, a mixture of several analytes. So a lot of people, has anyone heard of the peptide glucid? A lot of people check like mass accuracy. It's one peptide. Like, it's great, but the more data points you can get on the system performance, the better. So using a mixture is often preferred, particularly for chromatographic separation. You need to have at least 2 peaks to determine chromatographic resolution. And then again, if a company can establish that their product has been stable, validated, and so on, then that is the safer way to go. So on the left are some examples of what you can use. Certainly.
So you mentioned a couple of reagents, right? So HeLa digest and DSA, these things are we also use usually for testing the stability. How stable they are, like your experience from your commercial vendors?
Yeah, okay, so the stability of the HeLa lysate digest is quite good as long as it's above a certain concentration. That is one thing that is the most obvious effect related to stability, is if you have a sample that is very dilute in an autosampler vial, like a peptide mixture or even HeLa lysate, you will lose some of these low concentration peptides to the autosampler vial. And it's really annoying, because if you go through the dilution process, and let's just say your system suitability may be also related to a sensitivity check. So you might have things spiked in at a very low amount, and as they sit in the tube over time, they may degrade or disappear. Then that's not very helpful. So the HeLa lysate digest, in my experience, is very stable. I've used it for up to a month in the autosampler without really having any big deal. The peptides that I use are the ones from Pierce. They're called Pierce Retention Time Calibration Peptides, so PRTCs. I've had those in concentrated enough mixture spiked into the helolysate digest that I put in my auto sampler. I have an Easy Nano. And then I took that LC and I put it in my car. And then I drove to Ithaca, New York by way of the Adirondacks, so it was like a 2-day trip. And then I took it into a lab in Ithaca, plugged it in on a different triple quad, but the same model as mine, and the peptides gave the same results. And then I reversed the process on my way home, and the auto sampler sat in my car in like November for a weekend. And then I brought it back into my lab, plugged it into my triple quad, the same one I started with, and got the same response. So that is an awesome system suitability type standard. But yes, does that answer your question? Degradation?
It does, but I think, depending on which vendors we purchase these reagents, sometimes they are not always consistent.
Sure.
So that's where, and of course users have their own way of making these things, so if we make in bulk, that always may not work out, because
Sure. That is, and it's a bit of the chicken and the egg scenario where if you're trying to design a system suitability protocol and you don't yet have a sample established, you have to evaluate the sample while at the same time you're evaluating your system performance. And so when things begin to, for example, decrease in sensitivity, the question is, well, is it my sample degrading or is it my mass spectrometer? And so the things you could do in that regard is, well, if you think it's your mass spec, take the LC out of the equation, calibrate it, make sure it's passing all those check marks, and then hook the LC back up and make sure the LC is passing all of its, things that it has to do in order to be performing well. And if you don't see retention time shift and the mass spec is calibrated for mass and resolution, then you can begin to focus on the sample. But ask around. I teach a short course at ASMS that focuses a lot on system suitability, and we encourage everybody, like, if you have a method you run in your lab, give someone the details on it, have them run it in their lab. Everything should be really consistent, especially if you're using very similar kind of instruments. And so the system suitability concept for me really hit home when we did these cross-lab studies where we had no idea. We gave people protocols and sample kits and said, run them. And we didn't know, and we didn't specify the last time somebody calibrated their instrument. Triples, you don't need to calibrate that often, but it should be within 6 months maybe. And we didn't know age of electron multipliers and all these things. And so when we started getting data back, we're like, oh, this site is not doing so well, but everything seems to be okay. So we established the system suitability protocol, and then we were able to see variability between the sites for the same exact sample, same exact method, same exact everything. And we knew that, all right, the variability is now due to the instruments and not sample handling or anything else.
How often do you inject these samples?
There is no, so it's all, I wouldn't say there is a frequency that you have to stick to, but keeping in mind, any time you have a real sample where you're not sure of the quality of the data, then you have to back up to the last good system suitability that was acquired. So if you run your system suitability once a week, you are jeopardizing a week's worth of samples. You could get through a whole week, and at the tail end of that sample sequence, all of a sudden your data goes, and now everything up before that is in question, right? So I always try to bracket things. If you have a quick method, some people will do like 2 system suitabilities per 96 well plate. If you're running long nanoflow gradients and it takes a long time to run a sample, then like once every other day may be adequate. But just keeping in mind, if you bracket your samples and when you get a failing system suitability, everything before that is in question. So how do you assess normal system performance if you've never done this before and you need to start? The best place would be right after the instrument is installed. And if you already have your instrument or you don't know when it was installed because you just came new to a lab, start with the simple basics, tune and calibrate the mass spec, the LC, do flow rate checks, something as simple as you set the flow rate for pump A. And if you're doing high flow, you take the output before it goes into the column and stick it in a graduated cylinder and set your timer. And then you can tell if pump A is pumping what you think it is. If you're doing nanoflow, there are these little glass capillaries that you can buy from Fisher or wherever that have 1 microliter graduations on them. And you can take the part that goes into your nano column, take that tubing and just put it right up against there, and the liquid gets sucked in, and you set your timer. If you do 300 microliters per minute and you wait 2 minutes or 3 minutes, you get a microliter, right, approximately. So you can do these simple checks before you even start to make sure everything's running right. So then, once you have everything set up, you want to look at the variability of your metrics: peak area, peak intensity, fragmentation pattern, chromatography, retention time, peak width. Monitor them over a bunch of injections and make sure that those the percent RSTs are as low as you can get them. Anything above 10% really isn't so hot when all you're doing is injecting the same sample over and over again. Ask your vendor if they have a recommended system suitability. I happen to work for a vendor. I have a system suitability protocol I use, but I can't, like, in order for it to be out there in the public with application notes and stuff, it is a significant amount of work and legal crud. So when I talk to a customer that has a triple quad in their lab, I'll give them a method and I'll say, this is the sample I use. So ask your vendor, ask your salesperson, say, I need a system suitability, get me in touch with someone who can give me some details. And again, if you have, like, who here runs a Q Exactive HF? Okay, look at each other, talk to each other later about what system suitability stuff you've run, and then go back home to your labs and run them, and then talk to each other and, like, what did you get? What did you get? Because you should be getting the same thing, right? Even if the chromatography is a little different, the performance of the mass spec should be very, very similar. And so if you don't have 2 QEHFs in your lab, make a friend here, and this applies to SciEx, of course, too, and Waters, and all the other ones. So now let's just say you generated a bunch of data. How do you evaluate it? For thermo instruments that were born before the age of the Q Exactive product line, so LTQ Orbitraps, LTQ, LCQ, there was this executable file called raw meat, which you would bring your raw file in, and it would read all of the readbacks at every single scan. You can do this in Excalibur. You can go and see what the ion injection time was, what the measured like all these details are in there and retained for every single scan. This was just a nice way to pull it out. There are other pieces of software. I can specifically speak to Thermo because I work for them. Freestyle is some software where now you can generate plots across your gradient of injection time, temperatures of your analyzer, your vacuum pressure. All these things are monitored. And so you can get at that information using, from a couple of slides ago, this, they call it the NIST metrics because the scientists that wrote that paper were from the National Institute of Statistical Standards, and they have on that website a way for you to put your data through this kind of analysis. It gives you a lot of information out, so it may be overwhelming. More recently, there's through Skyline, Michael Berriman at North Carolina State has this piece of software called SPROCOP. And so you run your system suitability sample, it takes the information and then plots it out so that you can easily digest the data that you acquired. And it helps point to what may have gone or what may be going wrong if something's going wrong. And since then, hey, Lindsay, do you know, was this the basis for Auto QC?
Correct.
Okay, so
We'll be going into detail on this later.
yes. So Auto QC is linked to this SPROCOP, and they're all tied together with Panorama. And so we're presenting this because this is a Skyline short course. Panorama is a great way for you to just continually pop your system suitability runs up on a repository so that you can store them longitudinally. You can share them with your friends. So you QEHF people, you can do that. Database searches for peptide ID is a way. A lot of people use how many peptides did my yeast cell lysate get from my system suitability run, and a lot of people use a number, and that's decent. It's not the only thing I would do. Retention time viewer, so if you want to compare retention times from different labs, you can do it in Skyline. Before it was in Skyline, there was this program called Retention Time Viewer. You can access their Excel, even as something as simple as that, and popping data longitudinally in Excel and plotting it over time. And then, of course, check with your instrument vendor to see if they have software. So this, qualitatively, if you start generating this data, if something is really bad, you should be able to visually see, like, oh, that looks terrible. But, like I said before, it's not always this obvious. So it's important to define quantitative aspects that can discern a little issue from a big issue that's more obvious. So data processing in Skyline, things you can do is when you bring the data in, use this retention time plot. If you see a systematic shift in one direction, systematic shifts are bad in chromatography. If it wiggles around a little bit, that's also not ideal depending on the extent of the wiggle. Someone here the other day asked me what's an acceptable RSD or coefficient of variation for retention time, and I don't think about it that way. I think about it in terms of absolute minutes or seconds. If my retention time is changing by a minute, I don't care what part of the gradient it is in, that is unacceptable. I don't care what the percent RSD is, and if I take that change of 1 minute and divide it by 40 minutes because that's where it's happening to determine the percent RSD, that percent RSD isn't very big, but a minute is huge when you're looking at chromatography. So in Skyline, some helpful views if you are looking to start off manually, and these are all things that we've touched on previously, but this is the peptide area view. So if you have Skyline open and you've got your peptide peak area plot, if you right click and go to peptide view, this sorts, it has the peptides on the x-axis, and if you right click and go to order, you can sort them based on anything. I sort them based on retention time, so I know this one's always my early eluter and this one's always my late eluter, and then you can, this kind of acts like a histogram version of your chromatographic profile. You see the intensities of these peaks, you see the standard deviation, and you get a sense of what it should look like. Then, if you right-click and go to area CV, then you can see if here's the 10% mark. Not every peptide is going to be behaving in the same sample set below 10%. There are going to be some. This one has a high percent CV because it's a very low peak area. Same with this one. The ones with high CVs tend to have lower peak areas on average, and if you divide a big variation by a small peak area, that makes a large percent CV. So as long as you can look at it and make sense of what you see, that's very good. And then look at the peptide replicate view. So these are all different injections. You can sort them in order and look at the absolute peak area as well as the distribution of the transitions. So it's just, again, touching on everything we've talked about over the last couple of days, but using it in a slightly different way. So we did this cross-site comparison, and we initiated the system suitability protocol and started to see little issues pop up that would not have been obvious to us initially. So we had one site that ran this series of peptides. This was a mixture of 6 bovine proteins that were digested with trypsin. So semi-complex. It was definitely not a complex sample mixture. And we saw like decent variability for all these peaks. But based on looking at this data set and then the other sites that were bringing their data in, we saw like, wow, the peak areas for these is really, really low. That's unusual. Okay, well, let's look at the percent CV. Wow, if this is the 20% point, though, all of those later eluters that aren't really being well detected have a really high variability, and that's not good either. So we went to the site and said, hey, something may be wrong with your pump. Can you just check it? They were using an exigent pump at the time where you can do flow calibrations, and it takes a long time, and you have to do it for A and then for B, and then by a long time I mean like many tens of minutes. So if you're used to, you have to sit. It's not fun. So what they did is they checked the flow calibration, and they found out that the A pump was working fine, but the B pump, someone had entered in the wrong volume. for the amount of time the pump had been pumping to do the measurement, and so it was pumping too slow, which means these peptides were not fully being eluted from the column reproducibly and appropriately. So once they changed and checked, fixed their flow calibration problem, this is what it looked like afterwards, and this is the profile we normally see, and now they're percent CVs, so we're all less than 10%. And so this was a really eye-opening case, and we're like, wow, we got to get everybody running this thing. So here's an example of longitudinal system performance where we're looking at one peptide over a series of injections. This was September 19th, 2011, and this was September 29th, 2011. So here we were running system suitability at least twice a day, sometimes 3 times a day, depending on how it all went, because we just didn't want to have to keep re-injecting samples due to problems with the system. So this is what you would expect it to look like over that period of time. This is a second instrument looking at that same kind of peptide, and like this is just fine variability. This is kind of what we're accustomed to. But then you see this decrease. So that was not a good thing. And the person who was running it was like, oh, that's a pain. It may not be obvious when you go from injection to injection because the changes from run to run are very, very small. But when you look at it visually like this, you're like, let's fix this. So the instrument was vented and the source region was cleaned, and then the sensitivity popped right back up. That was the only change that was done. So when you start generating plots like this, and then you can annotate them like the dip was due to the source was dirty because it had been running for 6 weeks and hadn't been cleaned, and it was plasma, and then it was vented, and you can like keep track of things. Then when you start seeing repeating patterns, you can preemptively say, oh, well, I know after this many injections I start seeing a decrease. I'm going to stop it and clean it here so I don't waste all those samples. Because remember, these samples were bracketing like 12 hours of other things. This is another way. Analyst has software that looks at longitudinal monitoring. So you just plot one metric versus injection number or time, and you can get a sense of the system performance. And then here it's showing like the average and then one standard deviation of all of those data. Obviously, you'd like it if those standard deviation lines were narrower, or as narrow as they can be. This is doing that same thing in Excel. So if you don't have software that's ready to go, you can take the data out and plot it. So for discovery type instruments that are doing data dependent, you can run a targeted MS2 method where you've got a full scan and then you've got certain peptides or things you want to monitor that you just set up for them to monitor in the method so that you always are collecting data on them. I always spike my Pierce retention time standards into whatever background I'm looking at. So I don't consider that an absolute system suitability, but I do consider it something that I can track over time, all the time, and I can set up the method to specifically target them and get enough MS2 points across those peaks so that I can draw informed conclusions about my system performance. So this is another example of what it looks like in Skyline. This is over 1 year, and here you can see there are some times where you're like, something not right was happening. I wouldn't necessarily say delete these from your data, but have an explanation of why this isn't behaving like these. And then you can look at retention time over that period of time too. Obviously, if you put a new column on, your retention time is going to shift. And that's to be expected, but as long as you can make a note of it, then going forward from that point on, trust me, you don't want to be in the situation where you're like, oh, I've seen this problem before, and we fixed it, but what did I do? This is the kind of way that it will help, and using the notes feature in Skyline that are sample specific will allow you to keep track of stuff like this. So it's an electronic notebook. So for people that like to think about spectral counting or protein coverage as a good way for system suitability, this example is showing from the previous slide the peak areas for particular peptides that were being monitored. And for this injection, this protein, it's the same protein in all the samples, shows a really good sequence coverage. I think it's, I don't know where it is. Peptides shown in bold. red. Sorry, it's not clear up there. Oh, 6%. I can't find it. But the point is that coverage looks the same in all three of these runs, right? This run has the same kind of peptide coverage, the coverage or the one shown in red, as that one. But this one down here is like 4 times less intense than over here, but it's showing the same kind of detection and coverage of that protein. So there are some things that are not good metrics, particularly if you use them by themselves. If this was coupled with other details, perhaps it could be informative, but in and of itself, protein coverage or sequence coverage is not reflecting any change in the system performance where clearly peak area is. So for people that are like, how often should I run it? How should I run it? This is a nice flow chart in this paper, published in 2007, discusses the whys and the hows specifically, but this is just a nice flow chart where you set it up, inject heat solutions, make sure everything looks good. Is it performing well? Yes, start your batch. And then decision points on when you start seeing changes, what you should do. I like flow charts. So this was the first paper that came out from Berriman's group on S-Procop, where it kind of walks you through all the inputs in the process using Skyline to evaluate your hardware, and then the output plots that it provides. So it provides these longitudinal plots and very nicely visually shows you like average mean performance with 1, 2, and 3 standard deviations for the things being monitored, mostly peptides. And then the Pareto analysis shows you what is largely responsible for a change that may be seen and what is not, or less incrementally responsible. And then also mass accuracy plots, if you have high-res accurate mass. And so this used to be something that was just in Skyline, then it became QC My LCMS, I think, was a website you could go to, and now it's integrated with AutoQC, which works pretty automatedly, and Lindsay will go into details about that. But this is the kind of idea that you will get if you take the data. This is the view in Skyline of the peak area versus injection or QC number, and then this is obviously one peptide, but it breaks it down into all of the peptides that are being monitored, and you see that they have a similar shape. There's a trending. It's out of the performance, the lower threshold of performance acceptance. And so, here you would want to fix something, obviously, but more ideally, maybe over here is where you want to fix something. So if you start plotting it out, you can again begin catching things before they become dire, right? Because we don't want to wait on the engineer. You call and they're like, oh, we'll be there in 3 days. And you're like, I need you now. And some things are things that you are able to do, like changing rotor seals. It's really not that hard. I don't know, changing your solvents, right? How many people change their solvents once a month? How many people change it more frequently than once a month? Right? How many people change it less frequently than once a month? Oh, come on. I know, yeah, that's right. The recommendation for some of the true nanoflow pumps are to change them weekly. And I was like, who has time for that? And now I do, because I had too many instances where I just was getting irreproducible retention. Not only do I change them, but I make them fresh and I sonicate them to degas them, and then I change my solvents and I don't top off anymore. And I had enough engineers like, Sue, we can't come in and fix your stuff anymore. You got to do this because you're a thermo employee. I'm like, So listen to what they say because it can help prevent future downtime. All right, so in summary, set up a system suitability protocol that works for you and use it. It doesn't have to be complicated. Just remember, I am of the mindset that just do whatever is the simplest thing for you to do and start there. And if it needs to become more complicated, then you can add in those levels of complexity. Don't analyze your precious samples without having a system suitability in place, because ultimately your precious samples are going to generate data that will be making assumptions or conclusions or diagnoses on. And if you are not sure of the quality of that data because the system might not be performing right, then there is absolutely no point in generating those data. So make sure your system's running fine so that the conclusions you can draw from those data are valid. Use the system suitability to monitor changes in hardware, software, any changes at all. We used it a lot during some construction that was going on in our lab were you there during that time, Lindsay, when they put up the new building? And there's just like dust everywhere. And in one of the labs with the triple quads, we saw this increase in the baseline and it just kept going up and up. And it was because there wasn't the cover on the electrometer of the triple quad wasn't secured. And the dust and the particulates in the air were kind of going through as the HVAC turned on and off. And so in one run, you would see this baseline, and then it would hike up. I guess it would go this way, and it would hike up, and then kind of peter off. And you're like, what the heck is going on? And it was something that could be fixed. But we didn't want to see it in regular samples, so when we saw it in system suitability, you're like, oh, we fix this. And then keep examples of poor system suitability data, because usually these problems come back to visit you. And if you have a picture of a chromatogram, if you have, a peak shape or changes in mass accuracy or output from a data-dependent peptide search, then you can write down what you did to fix that problem and then keep it in a notebook. If it's something that occurs frequently, stick it on the wall next to your instrument. And it's very helpful, particularly if you have new people coming into the lab and you want everyone to be on the same page. All right. Here are a lot of papers. I wrote a paper on system suitability from all the things that we learned with the CP-TAC study. And so a lot of these are ones I found to be very good references. I don't like to do complicated things and I don't like to do repetitive things. And using these as resources helps to make things very simple and streamlined. So I would recommend, I don't know if I can get you copies of these now because I don't work in an academic institution, but surely someone can get them. We'll post them and make them available if you need them. All right, any questions on that? Who's going to go home and do some system suitability? Yeah, email me if you have questions, because I do have a system suitability protocol. I set mine up to challenge the performance of the instrument. So when a vendor says, this triple quad can do 500 transitions a second, all right, make it 500 transitions a second, and see if it really does it. Because then if in your method you're not doing 500 transitions a second, you're doing fewer, it should be able to handle it. And the same with data-dependent runs. If you have a certain number of MS2s you expect in a given run, whether or not they produce data, you want to make sure that they're actually being executed. And so there are a lot of people that have small variations on the same type of system suitability, and if people need pointers, what I have is not vendor specific. It'll work on any platform. So although they are thermal products, but you can get variations on those too.